I am recording the meeting. Okay, and now I will share. Hi, Gray. Everyone's here. It's a party. Okay, share screen 2. Okay. And that's a black screen and it's being recorded. Okay. Awesome. We'll get started in a few minutes.
Okay, welcome everyone to Liam's defense, right? So of course it's tradition in PhD defenses for the advisor to say a few kind words about the person who's about to defend. And so, this is Liam. He's been one of the really valued, really loved members of our lab. And I particularly love this image of him because this represents like a lot of the different sides of Liam. So on the one hand, Liam has grown into being a really exceptional scientist, but he's also a well-rounded person. We all know that he's one of the craftiest people you will ever meet. And so one of those crafts that he loves is knitting. And this is a sweater that he made that actually features our model organism. So we work on Drosophila. He captured some of the mutants. This is a curly-winged mutant that we do. He wore this to one of the huge annual Drosophila meetings, and I gather that he's a bit of a star in that. You're like, oh my God, did you make that? And so I actually posted this on Blue Sky, and I got in trouble with the knitting community because they're like, how can you post such a cool sweater and not give us the knitting pattern? And I went to Liam and I was like, what is this knitting pattern? Oh yeah, that's a no-no. You don't post a sweater that doesn't give like the ability to read it. That's quite like science. Right? So that's our Liam. That sweater was version 1.0. This is version 2.0. So this is actually this is a good primer for what he's going to show you. These are like our cells. This is what you'll actually see in some of his imaging, even the planar polarized patterns of this. But again, he's turned into a really great scientist. This is one of the nicest things about being a PhD advisor, is seeing kind of the growth and the continued work that he has done. That even included, for me, a special moment. This is a picture that Liam and Rashmi took when they got to meet my PhD advisor, Eric Wieschaus. Eric won the Nobel Prize there, so he's a bit of a science celebrity, and they got to hang out. It was very cool seeing Liam present his work to Eric, and maybe one of my all-time favorite photos ever was Eric was intrigued enough that he like grabbed a chair up and just to see him, right? You know,
I
like
wasn't even presenting that day.
staring at it and thinking about his work. It was nice seeing that there's a, you know, there's a complexity of thought to Liam's work that can intrigue a Nobel Prize winner there. Again, he's constantly pushing the limits. Sometimes it's the limits of what hat size. So yes, doing that. Also exploring the limits. This is when we actually arranged a bit of an ambush to the Van Engelenberg and Kushner labs. We challenged the goal. Then we had a total winner here. So remarkably, he had a game to say both a 299. 299. That's one pin short of a perfect game. But you can actually notice like the ball like hurt us. The ball just makes a sound. And you can kind of see other people in the bowling alley going. And yeah, it's just great. In one pin short. It's not to bring it back. Now this might be the painful part of the defense intro. One pin short of the perfect game on the very last one. We've seen many faces of Liam, right? We've seen baby face Liam. We've seen rocker Liam. We've seen I don't know Liam.
We're gonna die.
There. So it's been a pleasure seeing that. Throughout, he's maintained a fun atmosphere in the lab. He and Katie certainly formed, and I think Rashmi formed a dynamic duo of fun. So here they were at a conference, engaging in some hijinks. Again, there's our dynamic duo again. And he even introduced the next generation of students to how we lobotomize pumpkins in the United States here. So more hijinks, but probably the hijinks have peaked in something that I don't really have much control over in the lab, and that was the creation of the meme wall. So this meme wall started with a poster of Justin Bieber. Little did I know that that was going to be the entry drug to the meme wall. Pretty soon Darwin replaced Bieber's head. And from there, it just exploded and stuff. Sometimes I'm a little bit afraid to walk into the lab and look at the meme wall and what was there. One question has always been who originated the meme wall, and so I'm going to present some data in this intro. So our first piece of suggestive data is caught in the act there. Again, I think they're putting an exit sign over the window. Hopefully that's not yeah, that doesn't say what your experience doing.
I think there we were
was.
setting the suicide hotline.
Great. And I think we actually have the definitive data at this point. I think Katie and Liam and Rashmi definitely behind this. So he did have one nemesis during his PhD, right? The people on the third floor know what this represents, this blank section of wall. This is where our water fountain was. This water fountain has been 3 years in the replacement. You can actually see 2024, 2025. They're knocking that off. So Liam completed his PhD, hopefully, despite being dehydrated. And amazingly, just this week, all of a sudden, a new water fountain appeared. They actually still haven't installed it. So they're either going to do that this afternoon as like celebration,
That would be awesome.
or they're like, we're not doing this until Liam leaves. So, yeah. Just finish by saying, I think he could have a career in politics. I like this very professional looking photo of him and some of the lab members here. But I'll just close by saying thank you, Liam. Looking forward to your presentation here. And yeah, good to see you.
Oh, all right. Now switching over to my slides. I did it. That's right in Zoom. That's right here. Okay, cool. Oops. Okay, cool. Thank you for introducing me, Todd, and for embarrassing me with lots of pictures. I did wonder how many of those pictures of us being hooligans were going to show up in your introduction. I'm fairly certain I know everybody here. If there's anybody here that I don't know, one, why are you here? Secondly, thank you for coming. My name is Liam. This is my 5th year as a PhD student in the biophysics program here, and I'm excited to tell you about some of the stuff I've been getting up to these last 5 years. Okay, just a quick overview of what I'm going to talk about during my time today. Starting with an introduction of the lab's focus and the model system, and then I'll include some of the methods that the computational side of our collaboration likes to use for our data. And then I'll walk through some of the scientific outcomes described in the 2 chapters of my dissertation. Okay, so I'm a scientist, but I come from a family that's largely comprised of artists. And so every time I go home for a visit, I'm asked some version of, what do you do again? And this is a seemingly simple question, but my answer has changed pretty dramatically throughout the seasons of my academic life. So if you knew me in undergrad, look at that baby face. You might think of me as a physicist and a chemist, which were the things that I majored in college. If you're my younger siblings who have come to visit the lab a few times, you might think of me as keeper of the fruit flies. If you're a member of my lab or a collaborator, you might think of me as a software developer or someone with computational and analytical expertise. So to the question of which of these descriptions is more true, my answer is yes. But in all honesty, I consider myself mostly to be a cell biologist. And throughout my PhD, I kind of owned that identity a little bit more since the body of my work kind of lives in that world. And for the non-biologists in the room, when I say cell biology, you might think of a single cell on a dish, something like this nicely labeled drawing on the right that my sister Amy, who's a very talented 6th grader in the Russell Family Lab, drew based on her school notes when she took cell biology last year. And at this level of resolution in Amy's beautiful drawing, you can see that eukaryotic cells are these biochemical machines that are full of specialized organelles that work together to keep life going. And many people in the world of cell biology study cells at this level. So looking at those intercellular components coordinating with each other. But my interests are related to another fundamental element of cells, which is that they're often not alone. So cells in reality exist collectively in tissues like we see here. And in our collaboration, we're particularly interested in epithelial cells, which are the tissues that act as barriers in primary body tissues and organ linings. So these tissues have an interesting duality where they have to be dynamic and malleable enough to shape these complex organ plans and heal wounds and things like that, while simultaneously maintaining that necessary barrier function. So in addition to cells operating at an individual level, they also have to coordinate with their neighbors to maintain tissue homeostasis. So that's kind of the spin on tissue biophysics that my work kind of takes on. So you can imagine how tight cell coordination in tissues is especially important for complex topologies like this spiraling mouse cochlea here as an example. If the modeling processes that shape that spiral tissue are improperly regulated, then the organ will not function correctly. So the kind of larger scale question guiding a lot of my work is how do cells in tissues coordinate with one another to create and maintain complex tissue shapes? So the model system that we use to answer this question is the Drosophila embryo, which is where my crazy fly guy reputation comes in. And the fly embryo is a really great system for a lot of reasons. For one, the typical developmental timeline is really fast. Full embryogenesis, which is just the entirety of embryonic development, takes place in a little over a day. And you can see the entirety of that development here in this white light movie. And hopefully you can appreciate how many dynamic tissue shaping events have to occur in this very short amount of time. The short life cycle of the adult animal is also a benefit of the model system, since other developmental systems take months to develop when flies only take an order of weeks to get mutation lines going. And importantly to us as a microscopy lab, the embryo is transparent, so we can genetically insert fluorescent probes to watch live tissue shaping events in real time. So within fly development, the remodeling process that my work focuses on is an example of convergent extension. So this is where a portion of tissue grows in one direction while simultaneously shrinking in an orthogonal direction. And we focus on the extending germ band epithelium in the early embryo. And this remodeling process occurs at the onset of gastrulation. And in just over 30 minutes, this germ band epithelium will over double in length along the anterior posterior axis, which is the long axis of the embryo. And a few features that I'd like to orient you to before I play this movie is this is an embryo with plasma membranes fluorescently labeled. So each of these individual colorful polygons is a cell packed into this tissue. And at this point in development, there's a single layer of columnar cells surrounding the periphery of this three-dimensional embryo. And as you can see here, prior to extension, they're pretty regularly shaped and hexagonal in cross-section. And this dotted box here represents our imaging plane. So the actual movies we make on our microscopes are limited to that box. And I've false colored, well, I haven't, Ori White's false colored these rows of cells for ease of tracking neighbor relationships over time. So as I start the movie, you can see one of the especially striking features of the germ band extension fast phase that we study is that there are no cell divisions. So the majority of this convergent extension is achieved through simple neighbor exchange, where cells from these anterior-posterior aligned rows interdigitate with one another. And you can see that as the false colors of the cells getting mixed up, reflecting that these cells are directionally filing between one another. And hopefully you can appreciate how much this intercalation depends on each individual cell's ability to change its shape in a coordinated fashion with its neighbors. So zooming in a little bit tighter, our group performs analysis at the level of full cells and at the level of the contacts between cells, hopefully the reason for which will become clear as I walk through this intercalation model. So that twofold extension that we saw on the previous slide is really achieved by lots of smaller intercalary motifs all happening at the same time. So let me get my laser pointer on. So we have a group of 4 cells here labeled A, B, C, and D. And in between cell A and B, there's a contact, or cell contact rather, that is aligned with the short axis of the embryo. I'll refer to that direction as vertical going forward. I'll try to remind a few times. And this vertical interface is rich in myosin, which is a force-generating motor molecule. And that myosin motor shrinks this interface preferentially, creating a new contact between cell C and cell D, which were previously not touching each other. And that new contact between cell C and D then elongates orthogonally, stabilizes itself, and pushes cell A and cell B apart from each other along that long axis. So cell A and B that were touching before are not touching anymore. Cell C and D that were not touching are. So hopefully you can imagine how a similar process happening in lots of 4-cell groups allows for that macroscopic extension of the tissue. And these down here on the bottom are just some real cell images kind of illustrating what a T1 transition looks like in a living tissue context. Okay. So putting on my computational hat, capturing this germ band extension process with live imaging is an interesting kind of large data optimization problem. So that's what a lot of me and the computational people in the group think about. And since we're interested in dynamic changes in cell shapes and neighbor relationships, we want our microscopy to have a pretty fine-grained resolution, both temporally and spatially. And so we're often collecting an image volume every few seconds. Those image volumes will sometimes have 20 plus individual cross sections. For about 30 minutes, oftentimes more, to capture all of germ band extension, and a lot of times with 2 color channels to label cells and some other object of interest. So these data sets end up being really, really ginormous. We'll sometimes have projects that contain terabytes worth of data that need to be managed and processed. So this is kind of where this world of computer vision comes in. And I think a lot of my contributions to the lab have been optimizing our image processing workflows and implementing some newer computer vision sort of tactics to make our lives easier handling these huge data sets. So an example of some of the image processing that we might do, you can take this example image, which is again from a plasma membrane labeled movie. So each of these individual polygons is a cell. And this is kind of what the raw data straight out of a microscope would look like. So you can see the graininess from biological and electronic noise. And to make meaningful measurements of each cell individually, we need some way to label which pixels belong to which cell. So we use the watershed transform to accomplish this for cells. It requires a little bit of pre-processing. So we'll do a smoothing operation to kind of highlight the interfaces and darken the middles of the cells. And then the watershed mathematical operation itself is kind of like a local intensity peak finding algorithm. So this is a 2D representation of what that looks like, where we have these local minima catch basins, and then a segmentation line will be drawn at each of these local maxima. So when we do this on a 2D image, we get something that looks like this, where we have a skeletonized outline, and we have the positions of all of those pixels, and we can then use those as proxies for the bounds of each cell. And then we repeat that in every time frame in all of our movies. We can assign each cell a unique integer tracking ID, and then we can repeat the same process for every time point, and then kind of track each cell over time to see what it does, what its shapes are like, how it interacts with its neighbors, and how we can kind of quantify our tissue extension parameters using cell shapes. Okay, so that is all background and methods. I'm going to jump into the chapters of my dissertation. So chapter 1 is this project that me and my co-author Katie inherited from Tim, who was a previous postdoc in Dina's lab. This was really the project that I think we both kind of cut our teeth on in terms of learning how to code in MATLAB and how to think about image processing critically. And it was very much our introduction to convergent extension. So I learned a lot during this project, and the publication is now in the Journal of Cell Biology. So if you're interested in any of the stuff that I show in this abridged version here, you can go read the full manuscript there. Okay, so this project started out with a sort of paradoxical element of the intercalation model that I showed earlier. So imagine a cell lattice at the onset of germline extension that has this regular hexagonal geometry, looking something like this. All of the vertically aligned interfaces, or those so-called T1 interfaces, have an initialized contractile identity from a planar polarized distribution of that myosin motor protein. And previous work had sort of suggested that these vertical interfaces are specified at the beginning of tissue extension and that their identity is solely dependent on that initial specification. However, if that were the case, then once intercalation begins, you could imagine that all of those contractile interfaces would disappear and then intercalation would be done. And this would also theoretically produce these really weirdly shaped cells that are super elongated and aligned in a similar direction. But in our actual imaging, we basically never see that. So at these later time points, this is about 15 to 20 minutes into germline extension, we see more variety of cell size and shape, but we definitely don't see any sort of uniform elongation of cells in that direction, at least at the portion of the germline that we study. So this is inconsistent with the predictions of that theoretical framework if we have one initialization sort of process right at the beginning of tissue extension. And this kind of paradox is also supported when we quantify a population of interfaces that initially have that vertical orientation. So the initial pool of vertical interfaces do contract and disappear within the first 10 or 15 minutes. That's what this cumulative histogram kind of represents. And even though most of those interfaces have disappeared by 10 minutes, the planar polarized contractile behavior has not disappeared. So this heat map on the right is showing interface contractility. So that's the rate of change of the length of each of these interfaces, and it's resolved over angle. So this angle of 0 is those perfectly vertical interfaces that we would expect to be contractile and shrinking. And then positive and negative 90 degrees are the horizontal ones that we expect to be elongating. So this red blob in the middle and these blue blobs are the kind of expected polarized behavior that we have here. But if the only interfaces capable of contractility were the ones that are specified at the beginning of germline extension, we would expect a pretty significant drop off in this kind of red blob intensity here around 10 minutes, and we don't see that. We see persistence of those behaviors over time. So altogether, these findings kind of made us wonder if there is a mechanism that can dynamically reinterpret planar polarized identities over time. And spoiler, that is what we found. But to begin kind of probing this possibility, we focused on a population of interfaces that our typical T1 transition model largely overlooks, and that are these intermediate transverse interfaces. So these orange and purple ones. Since they're not vertical or horizontal, they're kind of left out of that model. And so the behaviors and fates of these contacts had never really been systematically investigated before this project. So to begin, we looked at contractile behaviors through this rate of length change of interfaces, and we resolved that again by orientation angle. So for the color scheme here, this red represents totally vertical interfaces and the purple represents totally horizontal interfaces. And when we look at angle bins in between those states, we do see a gradient of behaviors. So intermediate angles have intermediate contractile behaviors. And this is true both globally and over time, where we have kind of a peak in the difference in those relationships around the same time that we have a peak in intercalation rates. When we look at individual interfaces, we also noted that orientation angle is not static, which means that an individual interface will rotate throughout its lifetime. And you can kind of see that in this image here, where a newly formed T3 interface that's initially elongating starts to rotate and then starts to contract once it becomes vertical. So we also found that the direction of this rotation is preferentially towards those vertical orientations, and that is accompanied by that behavior switch. So as an interface rotates, like you can kind of see in this quiver plot animation giving a possible trajectory for one interface, as an interface rotates and becomes vertical, it contains that contractile identity associated with its positional change. So this phenomenon occurring for lots of interfaces could then potentially explain how planar polarized behaviors persist even after that initial population of vertical interfaces have disappeared. We're kind of systematically replenishing this pool of available contractile interfaces. And this contractile identity is also accompanied by increases in the actual myosin motor. So when we measure on an interface that's rotating, that is kind of being outlined by this white snake-looking thing. You can see that as the interface rotates and begins to contract, that red intensity also gets brighter, which means that we have more myosin. So this establishes that molecular planar polarities are also dynamically reinterpreted. It's not just a behavioral phenomenon. And this potentially emphasizes the importance of mechanical feedback loops as opposed to that gradient-based initialization of planar polarity. Okay. So we ended the paper with this cute little schematic. I still remember me and Katie sitting in the kind of lobby of the physics building. We were just like drawing cells on a whiteboard, and we were watching movies and drawing cells, and we were like, it's all there. And it felt like a big like brain blast moment because our model kind of proposes that this rotation is a geometric consequence of intercalation itself. So most kind of T1 transition models would end here where you have the newly forming horizontal interface, but it wouldn't really pay attention to what was happening to these connected transverse interfaces, which by the contraction and elongation kind of are required tissue integrity wise to rotate a little bit. So then you would end up getting those same group of 4 cells now having 2 newly formed vertical interfaces that could then do more rounds of intercalation. So this effect is systematic and global throughout the germ band and seems to persist pretty robustly over time. And I thought this was a really nice figure that we made together. So that's the rectification story. General takeaway summary type things. Interfacial behaviors exist on a graded continuum, and that is resolved by planar orientation angle. These transverse cell contacts that are neither vertical nor horizontal systematically rotate towards vertical orientations and become contractile as they do that. Planar polarized active myosin force generation machinery in the form of that myosin is required for this rectification. I didn't show that data, but if we kind of inhibit myosin, we also don't have robust rotation of these interfaces. And then kind of extrapolated consequence, this means that individual cells can participate in multiple rounds of staggered T1 transitions instead of just one. So this explains why we don't have super weird elongated cells and also why we have more than would be theoretically predicted in terms of total net extension. And then the initialization of planar polarized interface identity does not sufficiently explain the long-term behaviors. So we're thinking this is consistent with other work in our field recently that's digging into some mechanical feedback loop models for the persistence of planar polarity. Okay, so that was the first chapter. That was our first, we really like co-did that project. It was a good learning opportunity. Chapter 2 is on my independent project. That was my first author paper that is in review at development right now. I was really hoping we'd get the email from them this morning saying, you're in, but we're still waiting on that. And I have to give shout outs to Rashmi and Hugh, both of whom are here, who did some of the experimental work for this project. Huge helps. And yes, okay. So kind of jumping back a little bit into some of our methods. I think the rectification paper is a good example of how strong our like big data mindset can be even in 2-dimensional images. But recent interests in the lab and my recent personal interests are interested in a more 3-dimensional spin to our data. So as I mentioned, many of the works in the germ band are performed in kind of apical slices. And that's partially because a lot of the functional proteins in our cells are preferentially located apically. And also two-dimensional models of our cell lattices do a good job of predicting certain behaviors. However, in reality, our cells are columnar, which means they're really, really tall. So you can imagine like this image here is really representing a cross-section up here of these orthogonal views of our cells. But in reality, there are lots more layers that we can extract information from that might be meaningful in terms of fluid mechanics of our tissues. So that's kind of where my more recent interests are, is expanding a lot of our frameworks to this more three-dimensional mindset. And this three-dimensional framework also allows us to visualize cells still in cross-section like we typically would, but also in these 3D reconstructed views. And this means that we can move beyond thinking of each cell as like a pouch of homogeneous gel, and we can start thinking a little bit more about other subcellular objects like the nucleus. So I hope you'll agree that our curiosity in the nucleus is well motivated, given how much of that intracellular volume is dominated by this nucleus. And we know from other work that the nucleus is mechanically stiffer than the cytoplasm and other membranous organelles for that matter. And so we wondered if nuclei act as mechanical barriers for shear cell rearrangement. So that was kind of the focus of Noah and Rashmi's recent work that I helped wrap up when Noah graduated. So when we began investigating nuclear dynamics in the germ band, we identified two nuclear accommodation pathways. So the first is nuclear deformation, where cell shape changes provoke corollary nuclear shape changes. And this is represented here in this movie. You can see the cooler colors are more round and the hotter colors are more elongated. And we have more of those hot colors later on to show that nuclei take on more extreme shapes. Interestingly though, because the nucleus is stiffer than the rest of the cell, nuclei can't deform quite as extremely as the cells themselves. And so that kind of stiffness differential is seemingly overcome by axial movement of the nuclei along the apical basal axis. So this is where kind of the power of thinking about these as 3D objects comes in. So while in that cross-section, nuclei dominate the majority of the cell, along the apical basal axis, they don't quite span the entire height of the cell. They're about 2/3 of the height of these cells typically, a little bit less than 2/3. And so when nuclei move up and down relative to each other, they can unjam and allow for more efficient shear deformation. So you can see that from these kind of side view segmentation reconstructions, and also in cross-sectional images here, these empty cells represent cells that have had the nuclei move out of the frame, or out of our imaging cross-section rather. So with that kind of background context in mind, I'll introduce the genetic background that inspired the beginning of my project. So we became aware of this interesting genetic disruption where if you disrupt the maternal haploid gene, embryos interestingly do one extra round of cell division prior to the establishment of the germ epithelium. So a normal embryo would do 14 or would do 13 rounds of division, and this would be kind of the density. And these maternal haploid embryos do one extra 14th round of division. So we're jamming twice as many cells, twice as many nuclei into embryos that are the same size. And so we immediately wondered how these doubled cell densities would impact the mechanical conditions of the germ band, and also how those recently described nuclear accommodation pathways would translate into a tissue with twice as many nuclei. So that was kind of the genesis of this project. So here are some representative control and haploid embryos during convergent extension, where you can see the wild type on the left and maternal haploid on the right. And most obviously, these maternal haploid cells are really small. It almost looks like it's a control movie that's just zoomed out a little bit more. There's also a little bit less uniformity in cell size and shape, which again makes us wonder if the embryo is kind of struggling to accommodate this number of cells. When we directly quantify nuclear densities, we do see about a doubling. So this is consistent with that extra round of division truly producing about twice as many cells. So these images on the right, this is kind of the rolling window that I use to quantify how many cells per a certain square area we have in these embryos. And when we kind of corollarily, that's not a real word, when we measured the actual size difference, so kind of the inverse of that density, cross-sectional areas are approximately cut in half in all of these cells. So we see similar differences in cell perimeters, though interestingly, perimeters and areas don't necessarily scale uniformly with one another. If you have a hexagon that has half the volume, its perimeter would only be reduced by about 30%, and that's just like a geometric consequence. And we do see that consistency in our measurements, but it is also kind of interesting to think about the differences in like membrane availability in each of these cells, but that's kind of an aside. When we look at our columnar epithelial cells from an orthogonal side view like this, we do find that the maternal haploid cells maintain the same height as our control tissues, which in combination with that approximate halving of the cross-sectional areas means that these tissues are volume conserved as compared to control. So again, the embryos are the same total size, the same total volume. This indicates that there isn't significant tissue level buckling or curvature created by this doubling of the number of cells. And this also means that 3D cell aspect ratios are significantly altered to be relatively long and skinny. So ever since we have had this result a few years ago at this point, I've been calling these noodle cells because they're a little bit more noodly than our typical control cells are. So our condition is generating half cell volume, but with dramatically different 3D distributions. And if this difference makes a difference, mechanically, we would expect to see that difference captured in rates of macroscopic tissue extension. And we do indeed see that. So this quantification here, I am tracking the posterior end of the germ band. So when the tissue extends, it kind of goes up and around the back of the embryo. So when I track that tailing end, we found about a 60% reduction in extension efficiency in these maternal haploid embryos. So that's a pretty significant reduction in the speed of extension. And so for the remainder of this section, I'll walk you through some of the physical frameworks that we have found to be successful in explaining some of this defect, and some other kind of scaling related insights that these small celled embryos have given us. Okay. So I mentioned that interesting duality of epithelial tissues where they have to be dynamic enough to shape tissues and like heal wounds, but they can't sacrifice their barrier function. And so a lot of physicists like to think of these as kind of multidimensional fluid lattices, where you can imagine a solid-like fluid or solid-like tissue would look something like we see at the beginning of germ band extension, where you have very round, uniform shapes and a system that is dominated by elastic forces, compared to a fluid-like state where you would have more irregular shapes, a system that is dominated by adhesive forces. So kind of intuitively, I think about this solid state like if you had a Rubbermaid tub or something full of bouncy balls, as opposed to a tub full of water balloons. The water balloons would be more fluid like, the bouncy balls more solid-like. And then, of course, outside of our theoretical framework, our real images have the added element of time. So cell positions stay relatively constant in the solid-like state and then rapidly float in and out of frame in the fluid-like state. So with that in mind, we can use the geometric qualities of the cell lattice to quantify tissue fluidity. So the metric that I'm showing here looks at the relationship between a cell's area and its perimeter. In this case, it's the perimeter divided by the area square root, where a regular hexagonally dominated tissue that is kind of in that solid-like regime would have a value of about 3.7, whereas a tissue that's kind of entering a more fluid-like state would be at or above 3.8, which is the value associated with a regular pentagon. So in this quantification here, for the control curve in blue, you can see kind of the expected trajectory where the germ band is pretty solid-like in the beginning, and then as extension continues, becomes more fluid-like. But for the maternal haploid tissues in red here, you can see that we kind of stay within that solid-like regime even into late germ band time points. So this is consistent with our extension defects that we measured previously. And kind of work from our group and others in this field have focused on two primary explanations for a less fluid epithelium like this. One could be deficient myosin force generation. So you can see this movie on the left is an example where we inhibit myosin function and we just have a really paralyzed looking tissue where not a lot of stuff moves around very much. Or a solid-like tissue could be informative of like a more mechanically jammed tissue, which is related to that kind of nucleus-related story that I was telling you before, where if nuclei can't move up and down along that apical basal axis, they get in each other's way more, and we have a more solid-like tissue. So kind of starting off with that myosin force generating option, An element of epithelial cells that I didn't mention in the introduction is that they're oscillatory. So those myosin motors not only contract interfaces, but they also wiggle the cells around so that cells get bigger and smaller quasi-periodically. And that sort of oscillation-y motor is then part of what drives individual T1 transitions. And I mentioned that primarily because we quantified medial myosin force generation through those actomyosin-driven oscillations, and we generally found that there wasn't much of a difference between control and maternal haploid. So when we look at the mean intensities of kind of the medial middle of the cell pools of myosin, we saw, if anything, a slightly higher myosin intensity for the maternal haploid, but functionally the means are only a few percent different. So we didn't actually consider them to be functionally different from each other. And when I look at kind of correlative analysis of medial myosin in cell area and then cell area with itself, which kind of captures some of that oscillatory behavior, we again didn't really see much of a difference. So this is interesting for several reasons. One of them being it seems like that myosin force generation machinery operates fairly scale independently, even when you have cells that are half the size. Myosin force generation dynamics are about the same. But also it means that this explanation for a less fluid epithelium is minimally incomplete. So we kind of continued on to the nucleus. So starting with assessing how nuclear crowding could influence tissue fluidization, we made similar cross-sectional measurements of the nucleus that we did to the cells earlier. And sort of surprisingly, the nucleus does not seem to scale uniformly with the cell. So where we have cells that are reduced by, again, about half, nuclei at their midplane, so the thickest part of the nucleus, are only reduced by 20% in these maternal haploid embryos. So this is kind of interesting for several reasons. For one, it indicates that genetic content, since these are haploid cells that have half the amount of genetic content, that genetic content alone is not what controls nuclear size in our system. And then also, it seems like the material properties of the surrounding tissue are an important determinator for nuclear dimensions. So we continue to calculate a simple ratiometric relationship between the nucleus and the cell area. Again, this is at the nuclear midplane, or the thickest cross-section of the nucleus. And both control and maternal haploid tissues have a similar ratio early on, but the control tissues are able to clear up much more cytoplasmic space around their nuclei over time, whereas maternal haploid stays pretty much the same. So the amount of space being taken up by the nucleus does not change very much, which we wonder if that is kind of inhibiting efficient shear deformations of the cells. This tighter packing is also obvious when you look at nuclei laterally, again, thinking more three-dimensionally, where you can hopefully see in this maternal haploid image here how tightly opposed the nucleus is to a lot of these cell boundaries, where there's very, very little space as compared to the control, where you can see, a little bit of black background there. When we quantify this in these z-axis resolved area profiles, you can see that for control, this green curve representing the cell is pretty straight no matter where the nucleus is. But for maternal haploid, the position of the nucleus very strongly predicts the shape of the cell. So the cell has to bulge around the nucleus to accommodate its additional bulk. So when we quantify nuclear dispersion, which is that sort of second accommodation pathway that Rashmi and Noah described, we did find that the maternal haploid nuclei are significantly more staggered along the apical basal axis from the very beginning of germ band extension. So this kind of emphasizes the fact that because the nuclei are bigger in cross-section, they can't occupy the same kind of cross-section without causing, tissue breaking and bending and buckling potentially. And at first glance, this result might suggest that three-dimensionally, the maternal haploid nuclei are mechanically interacting with their neighbors less, that they're out of each other's way a little bit. But when we measure the distances between neighboring nuclei, so by calculating the minimum 3D distance vector between pairs of nuclei, we do find that on average, even though the maternal haploid nuclei are dispersed more, they're on average closer to their neighbors than the control is. Where you can see, again, for the control, we have this kind of progressive movement or progressive upward trend where nuclei get further from their neighbors over time. The maternal haploid nuclei kind of stay in this one uniform distance away from neighbors and seem unable to further unjam the tissue. So perhaps the maternal haploid nuclei are dispersed at the onset of tissue extension to try and kind of relieve some of those overcrowded packing pressures, but are unable to sufficiently accommodate for the increased density of cells. And we can also see this captured, this like inability of nuclei to disperse any further is reflected in periods of active nuclear movement. So here we used a mean square displacement based approach to identify periods of active movement in each nucleus's kind of apical basal trajectory. And in control, about 65% of nuclei have periods of active movement at some point during germ band extension, compared to only 25% of the maternal haploid nuclei. So even though these nuclei positionally are more staggered from each other, far fewer nuclei are actually actively translocating away from each other. And when we calculate the actual speeds of these nuclei, kind of similarly to the myosin intensities, maternal haploid nuclei might be a little bit faster, but the actual means are only a few percent different. So we don't necessarily consider that to be a super functional difference in the speeds. So in all, these nucleus results are consistent with nuclei being relatively trapped in their positions by these overcrowding effects of the tissue. And this asserts that our nuclear dispersion sort of mechanism by itself is insufficient in overcoming increased cell densities. Okay, so to summarize that second chapter, with doubled cell densities, the fluidization of the germ band is compromised fairly significantly. We did interestingly find that contractile machinery operates scale independently. So tiny cells, big cells, doesn't matter. There's some sort of uncoupling between cell dimensions and the biochemical actin force generating machinery. These haploid nuclei are larger than we would expect, and they scale non-linearly with cell dimensions. This causes the maternal haploid cells to be overfilled by their nucleus, and also asserts that DNA content alone does not control nuclear size, at least in an epithelium like ours. These increased cell densities generate more nuclear crowding despite upregulated dispersion, and kind of a bigger pie in the sky outcome that we put at the end of our development abstract is that cell densities seem to be evolutionarily tuned for compatibility with developmental timing requirements. Okay. So kind of like bigger pie in the sky kind of conclusions about why my work is important. These high throughput experiments that we perform in the lab, they allow us to surpass previous resolution limits, both in time and in space. And that allows us to reveal new and previously overlooked biology. I think the rectification paper is a good example of that, just in the sense that this T1 transition model that people have been publishing on for decades at this point had this very fundamental geometric consequence that nobody had ever seen before, just because they weren't looking closely enough. Also, fluid mechanics approaches, like the ones that we use here, can powerfully describe tissues, both during development, but in other contexts too, kind of indicating that collective cell behaviors can't be solely predicted biochemically. You have to kind of consider this more mechanical biophysical component. And also modeling tissues mechanically like this can allow for better understandings of human disease states that are kind of hijacking epithelial machinery, so stuff like cancer. And finally, kind of call to action-esque thing, fundamental cell and tissue biology research is still needed. We're still uncovering really basic foundational qualities of cells. And without this kind of laying the groundwork of foundational qualities of cells, we can't do any of the, more specified we can't dive any deeper into any more specialized cell types if we don't know the fundamentals of a model epithelium like this. Okay, cool. So those are the slides. Yay! This is my acknowledgement slide. This acknowledgement slide from the first time I ever did it has changed lots. Now I know how to cut pictures into hexagons on PowerPoint, so that's pretty exciting. I have to first thank my advisors, Todd and Dina. I originally applied to the physics program here, and Dina kind of pulled me from one of my interviews and asked me if I wanted to do biophysics instead. And then I met Todd, and the rest is history. So they've both been incredibly supportive of me, and I feel like have helped me grow into I think about science differently than I did when I came in. I think that's what you want to see, is that your thought processes change a little bit. And they've signed my paychecks, and that's great. Also, shout out to Skylar and to Kings, who are on my committee, both of whom I've had really fun conversations with about tech, about biology, about physics. So I'm very grateful to have the four of you in my corner as my committee. Hopefully I'll still be saying that later today. Have to shout out to my lab members. The current members, Prakriti, Katie, Prashna, Hannah Maria, who I leave the lab with you. I know you're going to do great things. I'm sad to leave, but I know that you're going to carry on. Super huge shout out to all the previous members. Tim and Noah taught me everything that I know about coding, which was very influential for me. Hugh was my undergrad. I could have him do experiments that I didn't feel like doing, which was really great. And he was just like a really fun guy to be around. Katie and Rashmi are my girlies. Like, I feel like us all being together in lab made it a really fun and meaningful place to be for grad school. So we get funded by the NIH for now, so hopefully that will stay the case in the future. And then I did the cutesy thing where I have more pictures for acknowledgements after the defense. So these are all like pictures of the lab and my cohort. This is the only picture I have of Todd and Dina together. And both of their eyes are closed. I just thought that was I thought that was very sweet. But with my cohort, we've gone to lots of conferences together. We've gotten up to tomfoolery. Everyone has been a good sport about me wanting to carve pumpkins every year, Very grateful for everybody. I'm grateful for my cohort because at the beginning of grad school, we all said that we were all going to do it in 5 years and we're all defending this year. So I think that's pretty exciting that we actually made good on that. I have a lot of really solid, awesome support system friends here in Denver too. Too many to name individually. So if you're on the slide, just know that I'm acknowledging you. One of those is my son, Fenway. I got him in my very first year of grad school. I recommend any new grad student in the room, get a cat. Get a cat, because if you have a really crappy day in the lab, you get to go home and there's a cat there. So that's pretty awesome. A special shout out to my best friend, Drew, who I have lived with for the entirety of grad school. I'm really glad we got to spend this season of our lives together. And my family are all here, mostly all here. My mom and my 6 siblings, I'm one of 7. And they all still live in Chicago. So everyone has been very supportive of me being far away. And I'm glad that I have such a supportive system to go back to. I did take screenshots of when you guys called me on my birthday last week. So that's what those are. Okay, and the last slide, and I'm not going to cry about this. My dad passed away in February this year, and he was such a dick. He was such an asshole, and he was so rude, and he was so smart, and he was so funny, and he was a computer scientist, and so a big part of the reason that I wanted to learn to code in the first place was because of him. And I think he died very suddenly, and so I don't think I was like ready to really like eulogize him at the time. But I hope you guys don't mind me taking a second to talk about him a little bit. So these pictures on the right, he came to visit me once because the Atlanta Braves were in town. And so he said, I'm going to come to visit you. And then I coincidentally found out the Braves were coming. I was like, interesting. But he came to the lab and he looked at some flies in the microscope. And he told people when he went home that I showed him a fly with 2 heads. I did not do that. I did not. He apparently told people that like all the time. What do your kids do? My son makes the fruit flies that have multiple heads. He's making shit up entirely. But yeah, I think he would have enjoyed to be here. I'm sad that he's not. But I'm glad that I can kind of attempt to carry on the positive parts of his personality in myself and in my work and in my future. Okay, that's all. Thank you all for being here, and I'm happy to answer any questions. I'm sweating.
Any questions for Lily?
Hi.
Hi, girl.
So the fact that the size of the nucleus and the maternal haploid only decreases a little bit, this is so tangential. So you probably
No, get into it.
But I'm curious if you ever looked at things like nuclear import and export and like nuclear force to see how that might change that.
Yeah, we
Or
didn't.
two on the.
For the stage of the project, it was a little bit more tangential than we wanted to go. We like really thought this was going to get published months ago, and then we've been going through the ringer with development. But that was one of the things on my mind. The other thing that I wanted to look at in terms of the nuclear scaling, apparently the nucleolus, I've heard people say it's so different. Apparently there's some justification or like evidence for that scaling more directly with gene content, at least in the fly embryo. So I wanted to look at that. We couldn't find a great marker for it. Import and export, we didn't super look at, but I wonder if that would be an interesting thing to look at in the future for sure. Yes.
Go back to your rectification thing, but I didn't see the wrong time.
That's okay.
From your rectification data, you show that mass and that makes that state of this probably explore the formation.
Right.
Know if they are also progressively being specific at the same time.
That's a good question. So for the rectification paper, we didn't specifically label bazooka. A different paper, it was that like methodsy paper for a segmentation, that Sega segmentation thing. And they also showed some amount of like a gradient of intensities and behaviors and stuff. And they showed that there is a bazooka gradient. We didn't directly measure that for the rectification paper though. And I think that was a Zalin paper. So I know that exists, but we didn't quantify it super directly. I am interested though, because it does feel like myosin and bazooka have that demonstrated antagonistic relationship. So I would expect we would see the time course of less bazooka being kind of like well correlated with the additional myosin. But yeah, I haven't directly measured that at all. Yeah, my bazooka flies wanted to die a lot. I don't know. I don't know what was up with that. Yes.
Cool. So nice talk. Congrats.
Thanks.
And I appreciate you also for legal advice.
I was gonna mention that to you. When she wrote lysosome, I was like, oh, she's gonna love that.
I have my questions similar to what Katie just asked, but kind of follow up. It looks like the gene comment is not decided in the third side, and also it's often not decided by the cell side.
Right.
Otherwise, we get shrink properties, right? So then I guess my question is, what is decided in the third side?
Right. And I think that's a great question. There's an element of, we thought about lamins potentially and lamin-like proteins if there was some sort of like structural architectural picketing thing that made them maintain a certain size. I never had I didn't do a ton of experiments like imaging lamins, so I don't have a great sense of what that outcome is. I'm kind of just feeling like there's a preferred 3D shape maybe just for the material content in our nuclei specifically. And so the maternal haploid nuclei want to still kind of maintain a similar like 3D shape factor to the control ones, even though the cells can be much more noodly. I don't have a great answer as to why that would necessarily be, aside from just kind of like a passive fluid mechanics idea of everything kind of wanting to be spherical, if it has enough tension. But yeah, that was something that we definitely left pretty open-ended to be like, maybe this could be something like that. We do have at least one person who's still interested in continuing to work on maternal haploid. They're doing some different stuff with it at different stages, but I wonder if some of that could be followed up on. Anybody?
Okay, you'll now proceed to the defense portion.
Thanks, everybody.
Assuming that goes well. 4 p.m. over the rotunda, there'll be pizza, there'll be cake, there'll be a little bit of champagne.
Party. Thanks, everybody on Zoom. I appreciate you being here. Yay. Hi. Sorry, I can't hear you. You can text me later. I can't hear you at all. Thank you, Tim. Thank you, Megan. And Cisco's here too. And Alyssa. Oh my gosh. It's a packed room. I've never had so many people in my Zoom room. Okay, I'm going to end the meeting now. Bye.